Welcome to the world of biological data analysis. In today's data-driven age, biology has transformed into a science powered by numbers, patterns, and computational tools. Biological data analysis is all about extracting meaningful insights from complex datasets, whether it's DNA sequences, protein structures, or ecological surveys. This video will guide you through the essential techniques used to make sense of biological data, from foundational methods to advanced computational approaches. Let's embark on this journey to understand how data analysis is revolutionizing the way we study life, starting with the basics and gradually diving deeper into specialized techniques. Biological data comes in many forms, each requiring unique analysis strategies. You might encounter genomic sequences, gene expression profiles, protein interactions, or even ecological population counts. Each data type presents its own challenges and opportunities. For instance, DNA sequences are long strings of letters, while gene expression data is often numerical and tabular. Understanding the nature of your data is the first step in choosing the right analysis technique. In the next scenes, we'll explore how these data types shape the analytical methods used in modern biology. Before diving into analysis, biological data must be cleaned and prepared. Data preprocessing involves removing errors, handling missing values, and standardizing formats. For example, sequencing data may contain ambiguous bases or technical artifacts that need correction. Preprocessing ensures that the subsequent analysis is reliable and meaningful. This step is crucial because even the most sophisticated techniques can't compensate for poor quality data. Next, we'll look at how data normalization and transformation further refine your dataset for analysis. Normalization is a key preprocessing step, especially for high-throughput biological data like gene expression profiles. It adjusts values measured on different scales to a common scale, making comparisons meaningful. For instance, in RNA-seq data, normalization accounts for differences in sequencing depth or gene length. Common methods include RPKM, TPM, and quantile normalization. By applying these techniques, researchers ensure that observed differences reflect true biological variation, not technical bias. Let's move on to how data transformation can further enhance analysis. Data transformation involves converting data into a format suitable for analysis. In biological studies, this might mean log transforming gene expression values to stabilize variants or converting categorical data into numerical codes. Transformations can reveal hidden patterns and relationships that are not obvious in raw data. For example, log transformation helps in visualizing fold changes in gene expression. With your data now preprocessed, normalized, and transformed, you're ready to apply powerful analytical techniques. Next, we'll explore exploratory data analysis. Exploratory data analysis, or EDA, is the process of visually and statistically summarizing your data. In biological research, EDA helps identify trends, outliers, and potential errors before formal modeling. Techniques like histograms, box plots, and scatter plots are commonly used. EDA provides a first glimpse into the structure of your data, guiding further analysis. For example, a box plot might reveal unexpected variability in gene expression across samples. Now, let's see how statistical hypothesis testing builds on these insights. Statistical hypothesis testing is fundamental in biological data analysis. It allows researchers to determine whether observed patterns are statistically significant or likely due to chance. Common tests include t-tests, ANOVA, and chi-square tests. For example, you might use a t-test to compare gene expression between two conditions. Proper hypothesis testing helps avoid false discoveries and supports robust scientific conclusion. Next, we'll discuss how correlation and regression analysis uncover relationships in biological data. Correlation analysis measures the strength and direction of relationships between variables. In biology, this might involve assessing whether two genes are co-expressed or if environmental factors influence species abundance. Pearson and Spearman correlations are commonly used, depending on data type. Correlation doesn't imply causation, but it's a powerful tool for generating hypotheses. Next, regression analysis takes it a step further by modeling relationships and making predictions. Regression analysis models the relationship between a dependent variable and one or more independent variables. In biological research, linear regression might be used to predict gene expression based on environmental factors, while logistic regression can classify samples as diseased or healthy. These models help quantify the impact of variables and make predictions. Regression analysis is a cornerstone of predictive modeling in biology. Now, let's explore clustering techniques for discovering patterns in complex datasets. Clustering is an unsupervised learning technique that groups similar data points together. In biology, clustering is widely used to identify gene expression patterns, classify cell types, or group ecological samples. Popular algorithms include k-means, hierarchical clustering, and DBSCN. Clustering reveals natural groupings in data, often leading to new biological insights. Next, we'll look at dimensionality reduction, which helps visualize and interpret high-dimensional biological data. Biological datasets often have thousands of variables, making analysis and visualization challenging. Dimensionality reduction techniques like principal component analysis, PCA, and TSNE simplify data by reducing the number of variables while preserving important information. These methods help visualize complex relationships and highlight key patterns. For example, PCA can reveal clusters of samples with similar gene expression profile. Dimensionality reduction is essential for making sense of high-dimensional biological data. Next, let's discuss supervised learning methods. Supervised learning involves training algorithms on labeled data to make predictions or classifications. In biology, supervised learning is used for tasks like disease diagnosis, species identification, or predicting protein function. Algorithms such as decision trees, support vector machines, and neural networks are commonly applied. These models learn from examples and can generalize to new data. Supervised learning is a powerful approach for extracting actionable insights from biological datasets. Next, we'll explore unsupervised learning in more detail. Unsupervised learning finds patterns in data without predefined labels. In biological research, it's used to discover new cell types, identify unknown species, or detect hidden structures in genetic data. Clustering and dimensionality reduction are key unsupervised techniques. These methods help generate hypotheses and guide further experiments. Unsupervised learning is essential for exploring the unknown in complex biological systems. Next, let's look at feature selection and its importance in biological data analysis. Feature selection involves identifying the most relevant variables for analysis. In biological datasets, many features may be irrelevant or redundant, which can hinder model performance. Techniques like recursive feature elimination and lasso regression help select important genes or biomarkers. Effective feature selection improves model accuracy and interpretability, making it easier to draw meaningful conclusions. Next, we'll discuss cross-validation, a method for assessing model reliability. Cross-validation is a statistical method for evaluating the performance of predictive models. In biological data analysis, it helps ensure that models generalize well to new data. The most common approach is k-fold cross-validation, where data is split into k-subsets and the model is trained and tested on different combinations. This process provides a robust estimate of model accuracy and helps prevent overfitting. Cross-validation is a critical step in building reliable predictive models. Next, let's explore the concept of overfitting and how to avoid it. Overfitting occurs when a model learns the noise in the training data instead of the underlying pattern. In biological data analysis, overfitting leads to models that perform well on training data, but poorly on new unseen data. Techniques like cross-validation, regularization, and pruning help mitigate overfitting. Recognizing and addressing overfitting is crucial for building models that provide reliable biological insights. Next, we'll discuss regularization techniques in more detail. Regularization techniques add a penalty to model complexity, discouraging overfitting. In biological data analysis, methods like lasso and ridge regression are commonly used. These techniques shrink less important feature coefficients toward zero, simplifying the model. Regularization helps ensure that models generalize well to new data and remain interpretable. By controlling complexity, regularization supports robust and meaningful biological discovery. Next, let's look at model evaluation metrics. Evaluating model performance requires appropriate metrics. In biological classification tasks, accuracy, precision recall, and F1 score are commonly used. For regression, metrics like mean squared error and R-squared are standard. Choosing the right metric depends on the biological question and data type. Proper evaluation ensures that models are both effective and reliable. Next, we'll explore visualization techniques for biological data. Visualization is a powerful tool for understanding and communicating biological data. Techniques like heat maps, scatterplots, and network diagrams reveal patterns and relationships that might be missed in raw numbers. Effective visualization helps researchers interpret results and share findings with others. For example, a heat map can display gene expression across multiple samples, highlighting clusters and trends. Next, we'll discuss network analysis in biological systems. Network analysis examines the relationship between biological entities, such as genes, proteins, or species. In systems biology, networks help model complex interactions and identify key regulators. Techniques like graph theory and centrality measures are used to analyze these networks. Network analysis provides insights into the organization and function of biological systems. Next, we'll look at sequence alignment, a foundational technique in genomics. Sequence alignment is a core technique in genomics and bioinformatics. It involves arranging DNA, RNA, or protein sequences to identify regions of similarity, which may indicate functional or evolutionary relationships. Tools like BLAST and Clustal Omega are widely used for sequence alignment. Accurate alignment is essential for tasks like gene annotation and phylogenetic analysis. Next, we'll discuss multiple sequence alignment and its application. Multiple sequence alignment extends pairwise alignment to more than two sequences. It's crucial for identifying conserved regions across gene families or protein domains. Tools like Muscle and MAFFT automate this process, handling large datasets efficiently. Multiple sequence alignment underpins many comparative genomic studies and helps infer evolutionary relationships. Next, let's explore phylogenetic analysis and how it reconstructs evolutionary history. Phylogenetic analysis reconstructs the evolutionary relationships among organisms or genes. By comparing aligned sequences, researchers build phylogenetic trees that illustrate shared ancestry and divergence. Methods like maximum likelihood and Bayesian inference are commonly used. Phylogenetic analysis is fundamental for understanding the evolution of genes, species, and entire ecosystems. Next, we'll discuss gene expression analysis and its importance in functional genomics. Gene expression analysis measures the activity of genes under different conditions. Techniques like microarrays and RNA-seq generate large datasets that require specialized analysis. Researchers identify differentially expressed genes to understand biological processes and disease mechanisms. Statistical methods and visualization tools help interpret these results. Next, we'll explore proteomics and the analysis of protein data. Proteomics focuses on the large-scale study of proteins, the workhorses of the cell. Mass spectrometry generates complex datasets that require advanced analysis techniques. Researchers identify and quantify proteins, study post-translational modifications, and map protein-protein interactions. Proteomics data analysis reveals insights into cellular function and disease mechanisms. Next, we'll discuss metabolomics and its role in understanding metabolic pathways. Metabolomics examines the small molecules, or metabolites, present in biological samples. Techniques like NMR and mass spectrometry generate rich datasets that reflect metabolic activity. Analyzing metabolomics data helps researchers understand cellular processes, disease states, and responses to treatments. Statistical and machine learning methods are often used to interpret complex metabolomics profile. Next, let's look at systems biology and integrative analysis. Systems biology integrates data from genomics, proteomics, metabolomics, and other sources to model complex biological systems. By combining multiple data types, researchers gain a holistic view of cellular processes and interactions. Computational modeling and network analysis are key tools in systems biology. This integrative approach is essential for understanding the complexity of life. Next, we'll discuss single-cell analysis and its transformative impact. Single-cell analysis examines the molecular characteristics of individual cells, revealing cellular diversity that bulk analysis can miss. Techniques like single-cell RNA-seq generate high-dimensional data, requiring specialized analysis methods. Researchers can identify rare cell types, track developmental trajectories, and study disease heterogeneity. Single-cell analysis is revolutionizing our understanding of biology at the most granular level. Next, let's explore spatial transcriptomics and its applications. Spatial transcriptomics combines gene expression analysis with spatial information, mapping where genes are active within tissues. This technique provides insights into tissue organization, cell interactions, and disease microenvironments. Analyzing spatial transcriptomics data requires advanced computational tools and visualization methods. It's a cutting-edge approach for studying complex tissues. Next, we'll discuss epigenomics and its role in gene regulation. Epigenomics studies heritable changes in gene expression that don't involve changes to the DNA sequence. Techniques like ChIP-seq and bisulfite sequencing generate data on DNA methylation and histone modifications. Analyzing epigenomic data helps researchers understand gene regulation, development, and disease. Integrating epigenomic data with other omics layers provides a comprehensive view of biological regulation. Next, let's look at microbiome data analysis. Microbiome analysis explores the communities of microorganisms living in various environments, from soil to the human gut. Sequencing technologies generate massive datasets on microbial diversity and function. Analytical techniques include diversity metrics, taxonomic classification, and functional profiling. Microbiome data analysis uncovers the roles of microbes in health, disease, and ecosystems. Next, we'll discuss ecological data analysis and its application. Ecological data analysis examines patterns and processes in natural environments. Researchers analyze species distributions, population dynamics, and community interaction using statistical and computational methods. Techniques like ordination, diversity indices, and spatial modeling are commonly applied. Ecological analysis informs conservation efforts and helps predict responses to environmental change. Next, let's look at time series analysis in biological research. Time series analysis examines biological data collected over time, such as gene expression during development or population changes across seasons. Techniques include autocorrelation, trend analysis, and forecasting models. Time series analysis helps identify temporal patterns, cycles, and responses to interventions. It's widely used in genomics, ecology, and epidemiology. Next, we'll discuss survival analysis and its applications in biology and medicine. Survival analysis focuses on time-to-event data, such as patient survival times or time to disease onset. Techniques like Kaplan-Meier curves and Koch proportional hazards models are widely used. Survival analysis helps identify risk factors, compare treatment outcomes, and predict future events. It's essential in clinical research and epidemiology. Next, let's explore machine learning applications in biological data analysis. Machine learning is transforming biological data analysis by enabling automated pattern recognition and prediction. Applications include disease classification, biomarker discovery, and image analysis. Algorithms like random forests, neural networks, and support vector machines are commonly used. Machine learning handles large, complex datasets and uncovers insights that traditional methods might miss. Next, we'll discuss deep learning and its impact on biological research. Deep learning, a subset of machine learning, uses neural networks with many layers to analyze complex biological data. Applications range from image recognition and pathology to predicting protein structures. Deep learning excels at finding subtle patterns in large datasets, making it ideal for genomics, proteomics, and medical imaging. Its ability to learn from raw data is revolutionizing biological research. Next, let's look at data integration and multiomics analysis. Multiomics integration combines data from genomics, transcriptomics, proteomics, and metabolomics to provide a comprehensive view of biological systems. Integrative analysis uncovers relationships that single-omics studies might miss. Techniques include data fusion, network analysis, and machine learning. Multiomics approaches are advancing personalized medicine and systems biology. Next, we'll discuss reproducibility and data sharing in biological research. Reproducibility is a cornerstone of scientific research. In biological data analysis, it means that results can be independently verified using the same data and methods. Practices like version control, detailed documentation, and open-source code help ensure reproducibility. Sharing data and analysis pipelines promotes transparency and accelerates scientific progress. Next, let's look at data visualization tools commonly used in biology. A variety of tools are available for visualizing biological data. Popular options include R's ggplot2, Python's Matplotlib and Seaborn, and specialized platforms like Cytoscape for network analysis. These tools enable researchers to create informative and interactive visualizations, making complex data more accessible. Choosing the right tool depends on the data type and analysis goals. Next, we'll discuss cloud computing and its role in biological data analysis. Cloud computing provides scalable resources for storing and analyzing large biological datasets. Platforms like AWS, Google Cloud, and Microsoft Azure offer powerful computing and storage solutions. Cloud-based analysis enables collaboration, reproducibility, and access to advanced tools without local infrastructure. It's increasingly essential for handling the growing volume of biological data. Next, let's explore ethical considerations in biological data analysis. Ethical considerations are vital in biological data analysis, especially when dealing with human data. Issues include privacy, informed consent, and data security. Researchers must follow ethical guidelines and regulations to protect participants and ensure responsible data use. Transparency and accountability are essential for maintaining public trust. Next, we'll discuss data privacy and protection measures. Protecting the privacy of biological data is crucial, particularly for sensitive information like genetic or health records. Techniques such as data anonymization, encryption, and secure storage are used to safeguard data. Compliance with regulations like GDPR and HIPAA is mandatory. Data protection measures build trust and enable responsible research. Next, let's look at the future trends in biological data analysis. Future of biological data analysis is bright, with emerging trends like artificial intelligence, real-time analytics, and personalized medicine. Advances in sequencing technology and computational power are enabling deeper insights into complex biological systems. Integration of wearable devices and mobile health data is expanding the scope of biological research. Staying updated with these trends is essential for researchers and practitioners. Next, we'll summarize the key points covered in this video. This video, we explored a wide range of biological data analysis techniques, from data preprocessing and normalization to advanced machine learning and multiomics integration. Each method plays a crucial role in extracting meaningful insights from complex biological datasets. Mastery of these techniques empowers researchers to make discoveries that advance our understanding of life. As technology evolves, so too will the tools and methods available for biological data analysis. Thank you for joining this journey through the landscape of biological data analysis techniques. Whether you're a student, researcher, or enthusiast, understanding these methods opens up new possibilities for discovery and innovation in biology. As you continue exploring, remember that the field is constantly evolving, and staying curious is key to success. We hope this overview inspires you to dive deeper into the fascinating world of biological data analysis.